So hello everyone, thank you all for coming. I'm Thomas Carroll, and today I'm going to give us an introduction to the Bioinformatics Resource Center. So I hope within the next 45 minutes, I can show you something about the Bioinformatics Resource Center, which should support you or help support you in your bioinformatics research. So to do that, I'm going to talk initially about who we are. So as Anthony said, the team has grown quite a lot recently. So I'm going to introduce you to the team. And excitingly, I can now tell you where we are. We've got our new digs, our new location. I'm going to show you a little bit of this. I'm then going to talk a little bit about what our role is at Rockefeller and what we should be doing and some of the difficulties in undertaking this role. And then importantly, I'm going to tell you how we can best support you in your own research through our pipelines and software, our training, and our custom analysis. And it's this last part, which I think we most enjoy and I feel is where we have the biggest impact at Rockefeller. Finally, I'm going to give a little teaser of what is coming next. What are we going to be deploying in the next year? And I hope that there will be some people here excited about this and maybe even would want to come and help us develop this. So as Anthony said, we started at the end of June, so I say July 2017, the BRC started, and this is when I joined to start this shared centralized resource for bioinformatics support. So we were just one, but now we are 6, and I really wanted to introduce you to the team right now. So here's me. Anthony's already given me a fantastic introduction. So I'll introduce the rest of our team. We have Jidong Liu. So Jidong joined us right at the very beginning of the team when we formed, and he came from Taiwan, and he brought with him all his skills from his PhD there. And now you'll meet JD. He's our senior bioinformatics analyst. You will meet him if you're doing any single cell analysis with us. You'll probably meet JD. So reasonably soon after that, about a year later, Matt Paul joined us from the Ercan lab where he was doing or finishing off his PhD. Matt brought all his skills with 3D genomes, but also you'll see a lot of work from Matt around our training, because this is something he was very excited about getting involved in, and he's really made an impact there too. Wei and Doug joined us way more recently. So Wei joined us just as we were going into the first lockdown. He joined us from the Green Guard lab where he was their bioinformatics analyst, and he was doing a ton of single cell work. So he's brought all these skills to us. You may hear first from Wei, because now we are working closer with the Genome Resource Center, and you'll get emails from Wei to deliver your FASTQ. So it's probably where you'll first come across Wei. Doug joined us June 2020, and we've been working quite a lot with Doug before he joined us. So he was finishing his postdoc in the Alice lab. So he joined us and brought all these skills in epigenetics. But you'll see lots of Doug's work when I start presenting our graphical user interfaces. And this is something totally new which Doug has brought to our team. Last but not least, very recently, Catherine Rosengagnon. Kate joined us just a few weeks ago, and Kate was finishing her postdoc in the Rice lab, where she was doing all sorts of interesting work. And so she's going to be bringing her skills in RNA binding protein analysis, so regulation of your transcriptome by RNA binding proteins, but also Kate had the difficult task of working in non-model organisms, so she's going to bring all these skills to the team too. So a really great bunch of people, and I hope you will find us and meet us. Oh, I'm missing a slide, sorry. Let me see if I can find that slide. Here we are. Okay, so as a team, we have experience mostly with next generation sequencing analysis. Hundreds, if not thousands of projects we have worked on with pretty much every flavor of next generation sequencing analysis. So this is our breadth of experience. But we also have some experience with proteomics, metabolomics, and we have some of these projects right now, and we have had some experience with image analysis. So we're really happy to support you in any type of analysis. We have a fair degree of experience now in bioinformatics software development. We have a number of packages in Bioconductor here. In terms of affection, we like R the most, but then also Python, JavaScript, a little bit of C++. But all the software we develop is based around the work we are either doing as our job, working in the bioinformatics core, or working directly with the users. So our software always comes out of the work we're doing. We try and make this publicly available. As you can see, we have a number of packages here, hopefully through something like Bioconductor or GitHub, so users can come in, pick up this code, software, make use of it themselves. Final thing, which I think we have a lot of experience in, is training. As Anthony mentioned, I was really excited about training before I came to Rockefeller, and I have a lot of experience. And now with Matt, together we've been putting together this really comprehensive training course here for Rockefeller. And I think we've run this every year since we've been here, a few months worth of courses, and I'm going to show some more of this later. So now, very excitingly, I can show where we are if you want to come meet this team. So we have been in flux between where we're based, but right now we are now based in Smithville Annex A33. Really excited to be here, really happy that Rockefeller's found us this location. This is somewhere here between the front of CRC as you move towards the new campus, the new buildings there. You'll come across us just off this kind of greenhouse area. And it's this wonderful little location, and we've got space for chairs, a little chit chat, discuss some bioinformatics. So I hope now we're coming back to the university. I hope we can meet here actually in person. So what are we meant to be doing as a team at Rockefeller? It's very simple. We are meant to be supporting bioinformatics analysis. Okay, broad, but it's very simple. And we aim to do this anywhere from project design through to publication. Ideally, we would like to be involved early in your project, like in project design, but I would say more than half of our customers, maybe 70% of our customers, are already deeper in their project when they come and discover or come and find us. They already have some analysis, they're already undergoing, and they want support to continue that analysis. And we are very happy to jump in and join in at any point during your own research. Up until the point that often people will come to us when they're in review, when they're trying to get this paper published, and they may need to add some additional analysis, and they will come back to the bioinformatics core, and we can help you just supplement whatever your analysis was. So feel free to contact us at any point to see if we can support you in analysis. So when I talk about analysis, though, that again, as I said, is broad. We really have a range of different analysis types we need to cater for at Rockefeller, and these can be simple to extremely complex. I've got this graph, which I actually made last year, so it's a little out of date. And I took this idea from another core who published it in 2015, but it's a really great way of summarizing what as a team we do. So on the Y here, we have the frequency, the number of times we do a project. And along the X, we have the different times we do a project, or the different types of projects. And we can see that on this very left-hand side here, we have our pre-processing and processing jobs, the type of thing we will do every day, RNA-seq processing. This is a little out of date, so probably now moving higher in the list with the single-cell RNA-seq processing, getting this count matrix together so we can deliver this to a user. So this is probably the most of our work, not the majority of our time, but the most of our work, pre-processing, processing. We would then have this middle ground here, and this is more standard downstream analysis, maybe from our pipelines or from data being brought from outside, but we will help users create things like heat maps, overlapping RNA-seq and ATAC-seq, maybe a UMAP nowadays, or something more complex than just default differential expression, multifactorial analysis, using your ATAC-seq and your RNA-seq. Okay, so we have a lot of this middle ground too. And then we have this long tail of work, which is, I think, the most important work for us to take on, and that is the custom complex analysis, clustering of neuronal signal responses, functional annotation of the genes in colony radiogram. These types of tasks we'll only ever perhaps do once, but they take a lot of time, they take a lot of attention, and they are also most interesting to us, and I think really what Rockefeller wants us to concentrate on. So Given this broad spectrum of work, I believe, and I have instigated, and this is what we're doing, that we need three pillars of our bioinformatics core. We need three kind of central tenets. We need software, pipelines, GUIs, or software and pipelines to handle this bulk of tasks here. Really don't involve too much human intervention, but they need to be done well and reproducibly. So we can have our softwares and pipelines maybe to handle that. We could use GUIs, graphical user interfaces, maybe to start to handle some of this middle ground, things which can require changing of parameters, some iteration. We also may or have been training and providing protocols and bioinformatics methods such that with as much support as they want from the BRC, users can maybe take on some of this work here and iterate through parameters and really get a better understanding of perhaps their data. And then our most important part, I believe, is our custom analysis. And this is how we take on this long tail of work here. So we need to be robust and reproducible and working as a team to try and cater for this long tail of work. In this long tail of work, things may not stay here forever. Functional annotation of colony radar ant, this may just become in the end functional annotation, non-model organism, become more popular, but there is always a long tail, custom complex analysis. So I'm going to talk through these three pillars and what we have in place to allow us to take on these different analyses types, and hopefully some of this can be useful to you. So the first thing I wanted to talk about was our pipelines and these graphical user interfaces. Let me make sure my graphical user interface is still open. Oops, sorry. Got a few slides here. Skip again. Okay. So first thing I did when I came to Rockefeller was be faced by large amounts of sequencing data generated by labs. So I sat down and I started writing our first version of what is now called NGS Piper. And this is a tool set to allow us to automate the common next generation processing, initial analysis, these types of tasks. Effectively, it can retrieve and process your data and do this reproducibly. It will perform some default standard quality control of your data. It will be able to tell you whether or not a sample looks bad compared to other samples. And we can perform some standard analysis. So if you give us group information, we can then do a pairwise comparison between every group. Nothing sophisticated, but the type of things we can do automated without knowing too much more information. From my previous work and working in other places, I have always just, found that wrapping up all this information in a deliverable report, which summarizes all this useful data, some of the analysis results, this is really important for users to be able to have in their hand and get an understanding of their data. So we concentrate a lot on making sure the report is intuitive, allows users to understand their data. And all that needed to be wrapped up in something which can scale from small to large data sets. We've run one or two samples, but we've also run thousands of samples at a time. And it needs to, of course, be highly reproducible, not just the software, but obviously the genomes as well. So if we want to take advantage of any of our pipelines, we now have high throughput sequencing pipelines for most common analysis types. You just need to contact us here at brc@rockefeller.edu. I'm going to show this email address a lot. And the process is quite simple. Once you've emailed us, we will meet with you. If we haven't worked with you before, we will set up an initial meeting. We will discuss what our pipelines are capable of, what type of data you want us to process. Once we've decided that this is capable, we're capable of taking this on. We will then supply you with a sample sheet, such as this one here. This is actually Matt put this together for us. I think Matt put this together for us. And then we'll ask the user to fill out simple things like where your FASTQ is or what the FASTQ name is, the sample ID, maybe some group information so we can do this pairwise comparison. It's actually got a little bit easier now with transferring data to the BRC. If it's external and you've worked with another resource center to generate your sequencing, you have to contact us and we can arrange to give this to us. If you're working with the Genome Resource Center in Rockefeller, because way is now commissioned by them to make the FASTQs. When you get your data delivered to you or upfront, if you know you're sequencing with Genomics Resource Center, just ask Wei to transfer it to us directly. We will pick it up and just begin the processing straight from there. Once we have run this processing, we will then have a meeting post-analysis. We will sit with you and we will go through the output from these pipelines, and we then discuss, is there anything else we can do now, next, follow on downstream analysis. Okay, so this is our output. This is output for one of our pipelines. Let's say we have them for ATAC-seq, ChIP-seq, single cell, RNA-seq, all sorts of different variations. One thing which we do consistently across all our pipelines when we create them is we make sure we find a good test data set of published data, which we can then recreate using our pipeline. So this means that we can really quality control our pipelines themselves by taking published, well understood results and making sure our pipelines can recreate them. So I'm just going to show you an example. My Zoom thing's at the top of my screen. I'll move it down there. So this is what the output report would look like from a standard here RNA-seq analysis. We also have them, as I said, for things like ATAC-seq or ChIP-seq. So in this case, just to show you roughly what it looks like, it is a report. The homepage tells you the types of files you're going to be given. These include things like signal graphs you can visualize in IGV. Importantly, not everything is just in this report. We do have Excel files containing your differentially expressed genes. That's explained here. Importantly, the data, how we tied your sample names to your FASTQs. This is important, how we've tied your sample names then to your cell lines or group information, and then what your reference data is. We have this link for IGV. I'm not going to show that today. And then we have the alignment statistics. So this is your quality control. And this is usually where we would go first with a user. We would open this up and we would start to look at their percent mapping, various metrics here, proportion mapped, but then how much is mapped into genes. And then finally, we have metrics like this distribution of signal over genes. Okay, so just a lot of quality control metrics, and this is what we would sit with the user. And it's fairly interactive, so we can quickly interrogate certain points. Once we're happy that the data looked okay, we will come to this page, and this is the default standard analysis we would do. But we can do a fair amount of standard analysis, reasonably straightforward way. Here we have our sample similarity heat map. Clearly, wild type is very different from everything else. And if we look in here, we can see something that wild type 3 really seems to stick out across this other source of variance. So, you know, this interactivity here is just allowing me to see which sample stuck out. So that was already very useful. We will do all these pairwise comparisons for you. So, you know, very stupidly, the computer will just compare every group versus every group in the order that is alphabetical. But this means that for every comparison, you will have a heat map so you can see how the genes have exchanged across your groups. Here we could tell that it's quite consistent, whatever changed tamoxifen versus ZHBTB, I think 53. The comparison is quite consistent across the other shRNA. We have this volcano plot, which is useful in its interactivity. I can immediately identify MIR30 was my highest upregulated. I worked on this paper originally, so I know that this SHPTB1, the backbone is MIR30. But if I wanted to find, say, PTB1, it's not obvious here. I can use this bit of interactivity just to filter down to PTB1, and I can see, yes, it's been downregulated. It's probably been reasonably highly downregulated. So yes, we have silenced that shRNA. On top of that, you have the rest of the differentially expression table. Yeah, you have links to IGV. You can look up what the gene is in gene cards. 30. And then you have gene ontology. And should we have run it, differential transcript usage. So this report, alongside the Excels and the deliverables, is really meant to allow users to jump in either with us or alone and start to interactively explore their results and get a good idea of what's worked and maybe what they could think about doing next in terms of downstream analysis. And so this is a nice fast pipeline we developed, interactive, but post actually running the workflow, you may decide this sample really sticks out, or this group didn't really get knocked down. So I want to remove it from my analysis, and I want to rerun the analysis, pull out the new results. Okay, that's going to be hard once we've already run the pipeline. We produce intermediate files, saved workspaces, which allow us to do this quickly. And should the user want to, they can also pick this up, and they can take the kind of post-process data and do a bit of work themselves. But with advice from our steering committee, and with a bit of elbow grease, we've really moved now to a more hybrid approach where we can use the workflow to do all the data intensive steps. So alignment, counting over genes, things where we're not going to be needed to enter any new parameters. And then we can have a graphical user interface, a website, which allows users to come on, interact with their data securely, form some quality filtering, regrouping, and then hopefully some initial exploration of that data. Then our workflow will still handle all this, but should the user want to, they can then go in and actually do a bit of this. So we put this together last year, and this is really Doug's work, Shiny NGS Piper. I think I've got the app open, hopefully. So it looks a little like this. You would get available, so I'm in the demo at the moment, you would only ever see data available to you, but Doug has added recently this ability to, I should probably hide some data here, the ability to share with other people in your lab, which is really fantastic. The app has all sorts of quality control, differential expression, and it's improved quite a lot since I presented it last year, or it's been made more parameters. And one I can do perhaps is I can make this blue, yellow, and set that. So because this is all post-processed, you don't have to wait around hopefully too long. I'm gonna, oh no, that wasn't too bad. And we can just do little extra bits on your data and really take it to customize it to exactly the figure you want. I'm not sure this is the figure I wanted, but it's what I asked for. What's really important about this whole system, and it was kind of a change in thinking for the BRC, is that once we've run all this, we want to obviously make it reproducible, not just in name, but we can wrap up everything we've done within a current analysis session, and we can save this as this really nice, neat little report, which contains all the outputs, every figure we've made, but also the code. So I'm going to show you that now. I've got one. This is going to be running here. You can actually review them directly in the page, but I've already downloaded one, and we'll see if this one completes in time. So these are really nice, neat little reports. We put a lot of attention into making sure they're readable, and they really talk you through the analysis. And then behind the scenes, you'll also have all the code you need to here set the working directory. So it'll click in JavaScript to tell you where this report is on your computer, set your parameters, and actually make some heat maps, etc. So you should be able to follow through this report, run every bit of analysis you require, as long as you follow through and set the working directories and set the parameters. And hopefully, if you can't quite get the figure out of our system, you have the code and you can manipulate it a little bit yourself. I think something which is really nice, and this is really kicked off by Doug, is this glossary which we've attached. And we're going to come back to this later. This glossary, this is open source. I should probably have a link. I can maybe show it at the end. But this sits on an open source repository, so anyone can contribute. And what we've done is we have words here. Doug's done our definition. So this will be linked in our file. And then this links both to a description of what FastQ is from our courses, but also really nicely is, how do you actually work with FastQ using R? So it's really nice. Oh, I've done something there. Yeah. So it's a really nice report coming out of this system. And this whole GUI I showed last year has now been in testing for about that time. We have over 50 users. I counted 61, but I'm not quite sure how many of these are really hopping on the app. But that's across 15 labs. And in total now, that's around 3,000 samples sitting in the app being tested and used. So, thank you to the 50 users who have been vocal, shall we say, and given us a fair degree of feedback, and really excellent feedback, which Doug has taken on. And there's been around 240 updates now to the system itself. And these include things like ability to share samples, and also perhaps really usefully, truly customize all these plots within the page. So I've rushed through this a little bit, but I'm going to show you a tutorial. I'm going to show you our YouTube page in a few slides. And this is the tutorial for the Bioinformatics Resource Center's RNA-seq analysis tutorial. You can really watch Doug step through every feature of the app, and then show you, all the fine details of this. And again, any feedback, we'd be really excited to hear. So on top of just our own app, which we presented last year, we have seen really a rapid expansion of people asking us to develop some of these graphical user interfaces. And I think we had one last year. We now have 13 apps in our repository across 6 labs or resource support centers. And these are being used for really interesting things. Quality control of assays. So you have an assay you're getting every time. You now want to make a report like the one I just showed you, maybe simpler, but you want it to be the same for everyone in your lab who's doing this. We've created some of these, and these work well. Contextualization of your results. So you have an Excel which has your results in. You want to go and append by gene name results from a database automatically. This is very easy to build for an app. But one of the most exciting things I've heard kind of come out of this is the idea that when you go to publish, your static image isn't really conveying everything you want to. And if you've been working on big data sets, that makes sense. Maybe you would want to use some interactive visualization to attach to your paper to kind of convey your data. And I got a really lovely example here, and I won't go into details because Doug and Edmundo are working on this. But I want to thank the Fischetti Lab just for letting us show. This is a really fascinating piece of work where we're taking compendium of all their work and all their sequences they've gathered, and we're able to put this into a phylogenetic tree, and then allow users to kind of click, zoom in, select samples, and compare. So this is commissioned from the Fischetti Lab, from Edmundo Campisi, who's a great collaborator. And I'm really excited to see this type of work starting to come to the BRC. So if you have any questions about our pipeline, software, or GUIs, please contact us at brc@rockefeller.edu, and we will make sure the right person gets in contact with you to discuss if you want to use our pipelines or you want to develop something new. So I now want to talk about our training. So we have quite a comprehensive set of training at Rockefeller now, and we've made sure that this really caters for different levels of training which people want from us. So a big portion of people who come to us, they just want to know what the files I gave them from our apps or from our pipelines really means. And genomic file formats course is one of our most popular courses. On top of that, we have people who want to cover this middle ground I showed in our frequency graph of tasks. We have people who want to do simple things downstream of maybe the data we've produced from the pipelines, and they want to take it, customize it, do some of their own analysis, and absolutely we want to support these people too. And the final group is also a big group, to be fair. And those are the people who want to know basic programming. Maybe they don't actually do any high throughput sequencing, they just want to know basic programming. But there's also a bunch of people who want to know exactly how our pipeline works. How do I align like you're aligning in the workflow? And we have made sure through significant effort that our training caters for that too. So we really go all the way through alignment, fast queue quality control, all the way up until analysis. Oh, we'll deal with that at the end, Marion. Thanks. Okay, so given we need all these different types of training, what I did when I came to Rockefeller, and as Anthony said, I had a bunch of training material in hand. I spent a long time in my first year rearranging this to make sure that our courses were not disparate, different courses, unrelated. We made sure that they built on top of each other, methods and data we saw in earlier courses was carried across into later courses, such that you may visualize something in the IGV course, and later we're going to show you how you align it from raw and visualize it programmatically. On top of this, something which is really important for all courses, and we've really tried hard, is that we need to make sure we use real world data. We can't just use toy examples, because otherwise when you leave our course and you go back to your table and start your analysis, you can't be sure it's going to work if it's just a toy example. It needs to be real world data. So we have a fairly comprehensive set of training now. This is the page. There's a lot of training here, which is great, but with more training material comes more weight for the bioinformatics core in order to maintain this. Right? And we want to do this well, but maintaining course material is time consuming. So we set about to try and build something which was going to allow us to get away with these things like software version changes, bioconductor updates every 6 months. If we actually make a mistake when we commit to the material, and if you're managing thousands of slides, it's easy to make one mistake. We need to have something which is going to make this easier for us. And taking advantage of GitHub and some of the new tools there, we wrote some internal software which now compiles all our material on Windows, Mac, and Linux. It generates multiple versions of the training material, and it creates our front page for our training, and that's this page here. So this is all automated from our templates. This is why every one of our training looks the same, and we can be sure that it has all the requirements you need to undertake the training. So the first thing we have is course integrity. We're a little harsh on the course integrity, I feel. We're a little strict on ourselves, but this is to check that it works on all versions, Mac, Windows, Linux, but also previous versions of the R, which is, I think, what catches us. And in this page, we show you how to set up. We can programmatically build the library dependencies. We have a link to download all the material. So if you don't want to stay on our webpage, you can get it locally. And then we can embed our training in these little sections here. We have our slides or our single page. Again, because we've done this programmatically, everything looks the same. And now it just makes it so much easier, not just for us to maintain it, but now we know how to do it, build them too. Nice feature, because we're using GitHub, is we can also have questions. So if you're not in class, I'll show that again. If you're not in class, we have this getting help, and this is on every single slideshow, every single course. And that get help will take you to a help related to the course you're on. And there we will have, Matt does a really great job here of making sure that we mark them as resolved, but we don't remove them from the page, so that people can come and see, I'm having an error installing Rio 2. How did they solve it? And a nice example here is Robbie, who we've worked with for a few years and was helping out the course there, and Robbie Williams from the Bursoy Lab. He answered the question. It's a public forum, so anyone can hop in and answer. And then Matt checked it over, related it to another question we had somewhere else, and then resolved the issue. So that's really cool. And that's been working really well for us since we've instigated that over the last year. A new feature we've added to the training, I can't mention this thing, this is failing. It's actually just failing on the old versions. But what we've added now is not just how to install R and the dependencies from the programs, but we actually developed a piece of software called Herper. Herper allows us to manage Conda repositories, hence external software, really easily. So now we can give users how to install the external software too that they need for the course. And then that will be further explained somewhere in here. I think Matt actually uses Herper to run. It's hard to find. There it is with conda env. So that's a Herper function. And also because we've built it into our page, it's actually getting tested too. So we're now not just testing the R, we're testing external software, making sure the results are correct. So that's great. And these pages, I think these are looking pretty complete now, but hopefully we'll find ways of improving them. Last thing I wanted to bring you to here is something which Matt did entirely on his own here. He's built up our YouTube channel, so we're YouTubers now as well. And this covers my graduate school bioinformatics course. We have material there, our talk, our training sessions. So Matt also ran some training sessions, the BRC tool guide. So this is Doug telling us how to use the tools, and then our RNA-seq analysis kind of flash course, which is I think what Matt wants to move to. If you come to this page, you know not where to start, I think it's a good idea as we set up a
Chief Human Resources Officer. Chief, stop treating humans like resources officer.
I'm not making any money out of these adverts, I assure you. But we have Matt, who will just introduce you to the page, explain a little bit about where to go and get you started. Well, I want to play a bit of it because it starts really nicely. I'll leave it for now. Okay, so that's lots of material on our training. How am I doing for time? Upcoming courses we'll announce later in the spring. Typically, we will do the graduate school somewhere in June, and Matt will also run, I think he'd probably do the full course again. He might decide to do flash courses, but we will make announcements later in the spring. For sure, though, we're going to concentrate now on doing at least a course with management, software management with Herper, but also we're going to start to try and pull out our Reticulate and Python courses, because this is becoming a necessity due to single cell. We use Python to analyze some single cell, so it needs to be in our courses too. Last thing I want to say about training, if you are interested in coming and actually running some training with us, we have templates available which can allow you to build the training the same way we built the training. Okay, so you just need to contact us and we can provide these templates to you. And I just want to point out that the VGL really kindly, they came and actually ran a genome assembly course, which was fantastic. And in that, I know Matt learned a lot about how to arrange these templates and help people build these courses. Okay, if you want any more information, when are we running this training? If you want us to run personalized training for a lab, or if you want to come and collaborate with us in training, please contact us again, brc@Rockefeller. So the last part of our 3-pillar core is custom analysis. And it's actually the simplest for me to explain, but it is the most time-consuming and I'd say most important part of it. And this is where either from data downstream from our own workflows or from data you've generated yourselves, we will work with you to try and find solutions, bioinformatics solutions to your particular hypothesis. And typically this is one-to-one consultation. I will usually bring a large proportion of my team to the first meeting. And this is so that, you know, between us, we can work out who is best placed to support you. But it usually comes down to one-to-one consultation. And over the years, we have had a range of different analysis types with Rockefeller. You certainly kept us very busy. Every flavor of sequencing in non-model organisms or even in organisms where the genome is being constructed still by other resource centers at Rockefeller. Lots of single-cell RNA-seq and attack in the last years. And then things like time course image analysis or proteomics, metabolomics. Really a broad range of analysis types. In all of this data, we try and make sure that we are obviously very reproducible. And to do this, we use things like dynamic documents, and these are markdown or flavors of this. which allow us to capture the versions, record all our code, and then hopefully share this with the user. So as simple as we can have our code in the document, the actual plot generated by the code. And by having this side by side, this allows the users to get a little bit more context on what these results really mean. And this just makes a better working relationship between us. Internally, we make use of Redmine to do our collaboration within the team. Redmine is just a system which allows us to log in, place analysis code associated to projects or users within projects, capture everything we need to know to recreate the project, as well as notes, descriptions. If you sent me an email, I might attach it to Redmine so that anyone else who comes on can read this too. And this is also where we configure and run our pipelines. With users, we try and work very closely one-to-one. And if we're going to be working to share code collaboratively over a long period of time, we try and take advantage of things like GitHub. And this is where we can use some of the same tricks you saw in the training. We can raise issues with each other, make questions, and use this kind of as a middleman to share our code, document, and raise these questions with each other. On top of being a fantastic home for us to collaborate through, nice thing about using these dynamic documents and combining this with GitHub is that we can usually turn this document into something like a PDF or an HTML, and we can actually attach that to your GitHub if you have one. And this is an example with Veronica from the Vossel lab. And in review, we came back and we needed to do some more analysis. We wrote this up into a PDF, a Markdown, into a PDF, and then we could attach this to her paper through GitHub. So it's not just a fantastic way of collaborating, it's a really good way of finalizing your output and attaching to your paper too. If you need some more support in custom analysis, feel free to just reach out to brc@rockefeller.edu. Either myself or my team will get back to you and we'll arrange a meeting where we can discuss exactly how we can help you. So in the last few minutes, I'm just going to close up on the custom analysis and then hopefully be able to show us something new. So one thing I wanted to say about this custom analysis is very rarely does stuff stay custom analysis for long. Typically, once something becomes more routine, we will look to either put it into our training or perhaps onto our pipelines, or most excitingly, we may try and write it up into some software. And a really fantastic example of this was with Doug, and this is how we first started working together. We had across a few labs, Fuchs, Bursoi, Alice Lab, same type of code, same type of analysis. And we built up such a repository of this code that we really wanted to write it up and make it useful to other people. So we took the code to make epigenetic heat maps. This isn't anything particularly specialist about any of these projects, but something generic we couldn't do with the current software. We wrote this up and made this publicly available through Profile Plot. So now not just us, but users at Rockefeller and more widely can take advantage of some of this code we produce. Follow on to that, I think is really nice and kind of completes the circle, is now that we have the software, we've now put together some training for this too. So our software starts to feed the training. And we've managed to maintain the use of real world data by actually going back and finding our collaborators we originally wrote it with. So in this case, I think it's Kvanch Versoy data. And we can just use a portion of this as our example training to show its use with real data. That's the end of our main section. And I just want to take a few minutes now to explain, what is coming next. So, well, as we met for our retreat, which was a weird affair because we actually came to university for our retreat rather than leaving university to go anywhere. But we came together, having been away for a while, and we started discussing what we wanted to do in the next few years. And one of the big things is we want to update our pipelines. So, NGS Piper, it's been around since 2017. Done a great job. It's been processed over close to 15,000 samples. This number just the other day. And it's been included, the output from this has been included in 25 publications. So it's been peer reviewed in some way too. And we've made sure that the methods we used in NGS Piper very deliberately, we've made sure all these methods are explained in our training. Having got together, we really want to try and now supplement our pipelines with additional description of actually what we've done. So really we want to be able to include a workflow description document, which is a self-contained tutorial explaining the workflow of the user's data processing. So a tutorial telling you what we've just done with your data. And the reason we want to do this is less this first reason, this call for availability of code. It will probably become a requirement. But really, we feel strongly that by providing users with a better understanding of our workflow, we can provide better context for the user to interpret the results. And this is something users have been doing for us since we came here. And I've blurred it a bit, but this is from Alexei Alislav. Users come to us and they give us wonderfully color-coded documents explaining your biology. And I think, the same reason you come to us to give us context, we should be working to give you context. But this turns out to write a short tutorial explaining the workflow turns out not to be so easy. Workflows themselves need to be complex. And the code becomes kind of unreadable because they need to deal with a ton of different situations. They work on high performance computing environments and they need catches for that too. Different inputs, different options. And this complexity can often obfuscate the relevant, useful parts of the pipeline to you. Your part of the pipeline is only a few portions of it, which is really relevant. And I put this wonderful cartoon here, but just to point out, I'm this guy on the right. People come to me quite regularly with workflows from previous institutions, and they say to me, can you recreate this? And scrolling through these workflows to find out exactly what was really done to the data. This part of the workflow connects to this part here. This is complex and very time consuming. Waste of time. Second part of this is, as I said, and very deliberately, we set about making sure our training covers everything in the workflow. So I could say, at this point, you can just read the manual, read all our training, and you'll work it out. But that clearly isn't a good enough solution. In the same way, the workflows need to be complex to cater for all these different parameters, inputs, environments they exist in. Our training is complex because we have multiple levels of understanding we're trying to give people. So it's very in-depth. So we need to think of something new. So we set about to rebuild our pipelines in a new system. And this system will now focus on providing additional in-depth documentation and explanation of the processing steps while giving us everything we had before. So I still want to get shiny NGS Piper. I still want my nice interactive reports, but I want this additional workflow document, which is going to describe how the bioinformatics core process your data. So I think we've done that now, and it's only just coming together, but I'm very excited, and we're starting the process of rewriting our pipelines, which is why this is what is coming next rather than what has happened. But I do want to point out some really nice features of it very quickly. And one thing is, we have worked very hard to make sure that we can deliver to the user not a complex set of conditional scripts, but a very succinct, easily readable piece of code which just relates to your use case. And we can do this by using this technique, metaprogramming, which is just using R to write more R scripts. But effectively, we take care of lots of the options upstream before we build the workflow. So we can create these kind of tailored, really easy to read workflows for the user, but still be standardized and reproducible. We have really made some effort to make sure that the workflows and the way we build them will be documentation description oriented. So it is inherently built into the way we build these workflows. So we shouldn't be able to generate a workflow which isn't highly documented. Off of that, we can take advantage of Doug's wonderful glossary, and we can now make this dynamic. So we can perhaps use that open source glossary, and rather than having it fixed in the page, we can use JavaScript to dynamically check the page. Do I have any of these terms? And then include them in like a glossary at the bottom. This is really important because anything which has HTML links and is static will die over time, because I will move where my training is. I may have to at some point, and then the link in your report will die. With this JavaScript, your links are always going to be correct. Finally, and these are probably the most, this here is probably the most difficult part, is we needed to make it mergeable. So we need to run part of our pipeline at one time, maybe a year ago, and that needs to be robust, but I need to go in and just take out parts of the output from, say, a workflow which generated our reference information. And I want to use that in my new workflow. And what's really tricky here was to provide this provenance of both code, but also the documentation and description. If I have my description in my reference building pipeline, I need that to somehow be carried across into my other pipeline, which is making use of it. That was probably the most complex. Finally, what we can do is add things like portable environments. So I've shown you we can use Herpa in our training to do things like install Salmon and allow the user to use this. So we should be able to make our tutorials on these workflows as easy to run, as easy to use, reproduce as our training is to run. So then this is what we've put together. I'm going to show you what it looks like here. And again, early days with this, but I'm really excited. So as you can see, it looks just like our training. That's deliberate. We want it to look very similar. We have the same getting help button. We haven't plugged this into anywhere yet, but in the same way that we can provide help for training, we should just be able to, this is going to take me to the RNA-seq one, but we should be able to provide a page where Rockefeller users can go and ask questions as well. In this report, we have, importantly, things like setting up your directories, building the Herpa environments. So this is going to build all the software you would require, build the libraries, and then everything in this first portion here, input parameters, we can show you all the details about how we've downloaded these files, created the reference information, and then zip this up. We also give you all the output as well. Anything which was generated by this code, we can stitch back together and give it in the output. So this is all the inputs. And finally, we would then move to the actual main processing, and this is the current pipeline. Hopefully you can see, this is really human readable. By putting a little bit of effort upstream, we've managed to make this really easily to read and hopefully follow. Last thing I wanted to show in it is our, if I can get it to load, is our dynamic glossary at the bottom of the page. So this is quite cool in that this is being read from this open source location. And anytime I come across a GTF in the file in my text, it will have been automatically linked to this table down here. And then this table will contain the description. If I didn't have GTF anywhere in my document, it wouldn't add this line. And then I can click here, and it will take me to my course describing exactly what this format is. So this is what's coming. I'm really excited to see this kind of come together. And I hope in the next year, you should start seeing this being attached to your Shiny NGS Piper outputs and your general outputs. So that's the end of the course today. It's quite long, I'm sorry. Just to give you some links to useful pages, inside Rockefeller, it's here. Our GitHub page where we have all our training and some links to our software is here. Our bioinformatics page is here. And I just want to, just point out that if you have any questions on any of this, please email us, brc@rockefeller.edu. All these slides I will put back onto our training website. And there will be a link there. Finally, I just want to thank Connie and the Genome Resource Center. We've been working with them much more over the last year, and they really are great support. I want to thank the Office of the CIO, Anthony, Jason, and Cecilia for all their help. I want to thank the team who are incredible, and as I've shown you, have been putting together lots of really cool things, hopefully to support. That's the end for our. If you have any questions, I saw some pop in. I'll try and answer them now.
Tom, thanks for the amazing talk. Really great stuff, and it's really exciting to see how the group continues to evolve, and looking forward to seeing NGS Piper 2 pretty soon. Thank you.
Thanks, Anthony.
Does anybody want to unmute themselves and ask a question, or just pop one into the chat?
I saw one question pop up in the chat I can probably address. So, Marion, yes. Are you adding proteome analysis training to the list? Not alone. So if we were going to do any proteomic analysis pipelines, we would want to work with Henrik. He's a real expert here. We are picking up some, and we've added, little GUIs to supplement results from proteomics analysis. But we are working a little bit with Henrik on this, on some of these GUIs. So I think it would, right now, if you wanted us to do anything, it would fit within the custom analysis service. But I hope in collaboration with Henrik, we can do something for proteomics analysis. No other questions? Thanks, Alison. Thank you. No other questions? Let me say again, thank you all for the support and for coming today. Find out any information on our website. Please contact us if you have any questions. Thanks, Lewan. Should we close up?